If you are unfortunate to have a heart attack, billions of cardiac muscle cells will be lost in your heart. And because the human heart cannot really replenish these cells with new heart muscle cells, the default mechanism of repair is by depositing a scar. This scar is actually needed to start with to avoid ventricular rupture, but the problem is that because this scar is permanent, will lead to heart failure. And this means that the heart cannot really pump blood as efficiently through your body as before damage. So if we could understand how this scar is being formed and deposited, we could potentially modulate it and avoid heart failure in cardiac patients. We use 3 different ways of studying heart repair and regeneration. The adult mouse heart, which behaves very similar to a patient that has a heart attack in the sense that it cannot regenerate the heart, and the default mechanism of repair is by forming a permanent scar. The neonate mouse, which can, with very minimal fibrotic deposition, repair the heart up to 7 days after being born, and after that something gets switched on or switched off and it loses that ability. And the zebrafish heart, which can repair and regenerate the heart up to adulthood. So we wanted to understand if macrophages, which are these cells that are usually associated with inflammation and fighting infection in our body, could or would behave in a different way at the transcriptomic level in these different settings. And we were very surprised to look at macrophages in both fish and mouse and see that they were upregulating molecules that actually make part of a scar, in particular collagens. We found this observation quite striking because the current dogma in the field is that myofibroblasts are the sole cells depositing collagen in the damaged heart. And with our work, we could find and we showed that indeed macrophages not only upregulate collagens after injury, but can also export these collagens to the extracellular matrix and deposit it in the injured heart. So with this work, we found the evolutionary conserved new role for macrophages that we believe could also apply to the human heart. To effectively repair the heart, broadly speaking, you need 2 things. One, you need to modulate the permanent scar into a transient scar. And 2, you need to replenish all these heart muscle cells that have been lost by injury. So our study can really help in the first part of addressing the first part of the problem, as we identified macrophages as a new player in depositing this scar. But I would like to stress out that before we are able to move to the clinic and be able to help patients that have suffered a heart attack, we really need more research and really trying to understand deeply what is the mechanism by which macrophages can indeed contribute to this scar, which molecules are involved, what is the relevance of this scar. So more fundamental science is really needed to be done.